Good morning, everybody. I'm Adele Krach, and I'm from Food Focus. And it's wonderful to have you present this morning. And we want to thank the Dairy Standard Agency for sponsoring another webinar in the series of webinars that they've been hosting this year. The DSA is working very hard, Jacqueline and her team, to ensure that they provide you with information to ensure that you can with them work to look at the quality of all milk products. And this topic this morning is yet another topic that will provide you with information to ensure that we can make the products and the milk products that's out there to a standard that is safe for our consumers. Our topic this morning is a very interesting topic. It's about E. coli from a One Health perspective. In this webinar, we will focus on some of the significant aspects of the microbiology, the epidemiology, and the host pathogen interactions of E. coli in animals. We will also look at it from a One Health perspective, and we will address animal and environmental reservoirs and proposing interventions targeted at pathways of transmission to optimize effective prevention and control measures. Now, we've got Dr. Chris, that will be presenting this morning. And Chris, if you can please put on your camera so that the audience can see you. There you go. And I'm going to put my camera off, and then I will introduce Chris. Chris is a veterinary specialist, and he's worked for almost 40 years within the private practice, public and corporate spheres, beginning his career as a veterinarian and later as a veterinary specialist in a veterinary practice. From 2000, he moved into the corporate arena with one of the leading animal health companies in the world, servicing South and Central Africa. And in 2015, he joined the MPO, and there his role as the executive head was to include and engage with stakeholders at prominent levels, driving the identification of value-adding products and services and research and development of the needs for the sector and their participation. In 2021, he embarked on a new venture as a specialist, and he's consulting for the FAO and other pharmaceutical companies, and then for the South African Veterinary Association and various other groups. Chris, thank you so much for your valuable time. We really appreciate it, and we are looking forward to your presentation.
Adele, thank you. I trust that everybody can hear me, and thank you again to be part of this meeting. You know, it's a joint venture between Dairy Standards, playing a very, very important role in the dairy industry, and then also Food Focus. So thank you for the opportunity. You know, Adele, as you said in the in the introduction, I would like to look at E. coli or Escherichia coli, as it's usually Escherichia is a difficult long word. We usually talk about E. coli. Now, E. coli was first discovered in 1885, it's almost 140 years ago, discovered by I think it's a Swedish doctor, Theodor Escherich, so that's where the name Escherichia coming from. So Escherich, Dr. Escherich, discovered this in 1885. Originally, it was known as Bacillus communis coli, and later on the name then that changed to Escherichia coli. Now, I think we're all aware of the name, and several times in discussions. The name, when we discuss dairy and when we discuss disease and so on, especially when you look at neonatal, the young calf on a dairy farm, E. coli is one of the names that will be frequently mentioned. So we're all aware of the name, and I think we all have met this little bacteria once or twice in our life, and I think some of us will remember that meeting. It's not a nice thing to have E. coli gastroenteritis. But yes, it can cause a lot of other diseases also. So what I'm going to do today is, to focus on specifically in the first part of the presentation. I will be focusing on specifically on From a veterinary perspective, because I'm a veterinarian, I would like to give you a little bit more detail there and then address all the things that can cause scours or diarrhea in calves. And then, in the second part of the presentation, then I will look at the, as Adele said, on the One Health, or look at it from a One Health perspective. And then, the role at land and then water and, soil and so on, and then the animals on the land can play. And then also the role of humans and the role of the pathogen itself. If we look at, the infectious scours or diarrhea agents in cattle, I think we can loosely divide it into bacteria, viruses, and protozoa. Now, the bacteria, the important one, what we're going to discuss today, Escherichia coli, or then abbreviated as E. coli. We get the different serotypes of Salmonella or Paratyphus, as we sometimes call it. And then certain forms of Clostridia, Clostridium perfringens type C, specifically, known to cause diarrhea and eventually mortality in calves. And then very important, the viruses. Usually the viruses and the E. coli, at the very early stage, Rotan coronavirus can play a role together with E. coli. It's usually the virus that will come in that will damage the villi in the intestine itself. And then as almost as a secondary infection, the E. coli can come in. But the E. coli can also be a primary infection and cause a lot of problems. And then the protozoa, I think Coccidia has always been there. Cryptosporidium, when I qualified as a vet in 1982, I think we didn't even talk about Cryptosporidium. Now it's on everybody's lips, and we're talking about it, and it's causing havoc in dairy enterprises, young calves, in beef calves, and then also in sheep and so on. So there's a lot of research ongoing on Cryptosporidium, but the two, also E. coli and Cryptosporidium, usually can be associated with some form of immune suppression. Immune suppression in humans, as an example, with your HIV in humans, but then also in animals. There's certain viruses, your BVD virus, your enzootic bovine leukosis and so on, and then also your herpes viruses, the IBR as an example, can cause immune suppression. And then, as a secondary, almost as an afterthought, E. coli will come in and then cause, severe problems there. So this is a summary of, I think the important diarrhea agents that can cause problems. Here's a little bit more expanded, so what I would like to focus on today is the E. coli. You can see the age of onset there. I think it's important also if you want to diagnose diarrhea on a dairy farm. Cryptosporidium usually a little bit later on, Coccidia a little bit later on, Salmonella usually after just the important word there is usually, because by biology, there's usually rules in biology, but usually it will be rewritten in the textbook. But the important one is E. coli there, and you can see most often between day 1 and 7. I've seen calves that actually it's born the morning and the next morning it's dead, and then when you go and do a post-mortem, you find that it's E. coli. And then you go back, it was born in a very dirty calving camp, got infected directly during the calving process, and then, that calf will then go down with a highly virulent E. coli and will be dead in 24 hours time. So that can happen. And that's usually associated with, a yellow to a white feces, projectile diarrhea. It's a severe diarrhea that we do get in E. coli infections in calves. Okay, so that is a summary there. I think if you look a little bit more detail, there's a nice picture of E. coli on the right-hand side there. But this is a common cause of bacterial enteritis. What happens after ingestion is usually through the oral. It will be ingested, via the mouth. It attached to the cells of the lining of the small intestine, the small little villi in the intestine there. It will go attach there with that little fimbriae that you can see there. And that's where all the problem starts. Then it starts proliferating, and then in this whole process, it starts secreting toxins. I will allude a little bit later on to the toxins, because that's a part, the important part of the pathology itself. And then in the process, the intestine then, or the animal then, immune system then recognize there's a problem. And then this whole flushing process, the diarrhea then ensue, and this leads to malabsorption. severe very quickly dehydration. So treatment of E. coli coughs, first of all, look at to restore the dehydration. The electrolyte imbalances, metabolic acidosis can happen, and then a potential death is sometimes one of the problems. So this will, the so-called intestinal form, and then when it gets absorbed, I'll show you a picture later on, when it gets absorbed through the intestinal lumen and it ends up in the bloodstream. This is the so-called Coli septicaemia. We frequently see it in small little chicken. In poultry, we see it. We see it in pigs, but also in calves, where so-called Coli septicaemia can happen. And that can cause, whichever organ it's going to, if it's going to the kidneys, and it can cause a nephritis. If it's going to the brain, there was an outbreak of E. coli septicaemia now recently in a flock of little lambs there. And about 20 or 30% of them died because of a meningitis or inflammation of the membranes in the brain because of an E. coli septicaemia. So this, this is not something that you need to play around, and it's been around, as I said, it's been around for more than almost 140 years compared to COVID, that's now been around, I think, quite a long time, but we only learned all the consequences in the last 4 or 5 years. So yeah, this is a problem. To go a little bit deeper, I'm not a microbiologist, but just to take you to that, this is a gram-negative facultative anaerobic bacillus. So it can be, in an aerobic environment, but it prefers an anaerobic environment. As I said earlier, it's a normal inhabitant of the lower intestinal tract of adult cattle. I think if we go and take a sample, fecal sample of 100 animals or 100 humans, we will find that there's E. coli somewhere, in 20 or 30 or 40% of those animals. But it's not necessarily that it's pathogenic. So not all E. coli can cause disease. And I think that's a very, very, very important thing that we need to recognize. As I said, the clinical onset's, 1 to 2 days. And this is usually, as I also said, found in combination with scours pathogens or your diarrhea pathogens. So that's why it's so important, if you look at a vaccine, specifically in cattle, it should contain rotavirus, it should contain coronavirus, it should contain E. coli, and then also your clostridiums, to make sure that you get that combination there. And I will refer back to the vaccines a little bit later on. Now, if you look a little bit more into the microbiology, the international serotyping scheme there on the left, I've highlighted it in green there. This is on, based on the following antigens, the so-called O antigen. This is the polysaccharide portion of the cell wall, and then you get the capsular polysaccharide, the K antigens, and then the H antigens, and you also get in sheep, especially in sheep, you get the F antigen. So I think it's important, that we know who is the enemy. If we identify E. coli, it's good to identify it, but then we need to go and look and see, do we actually have these antigens? And, in the last 140 years, and I think in the last 10, 15 years, we expanded, if you look on the right-hand side there, the E. coli genome. That's just E. coli K12 there. You can see at the bottom there, the genome size of Escherichia coli 0157. The base, so-called base pairs there, more than 5.5 million base pairs there. So this is what we're busy looking at. Yes, it's good to have the identification on the left-hand side, but we went to the, there's a lot of advances in microbiology, and this is where we're going to, to look at, specifically at the virulent factors, but then also to look at, in the development of vaccines, we will use this, but then also in the treatment, we can identify there could be certain markers on this genome that will tell you this organism is resistant to this or that antibiotic, and then we can actually introduce a new antibiotic. So if you look at the types of E. coli, I think this is also important. I'm not excuse all the detail on the slides. You don't need to read all of it now, but I've put all the detail there so you can go and read it later on when you have a relook, hopefully a relook at this whole presentation. You get the enterotoxigenic E. coli, and the important one there is the attachment K99F41 that attaches to the intestinal lining, and that produces the toxins, so-called abbreviated as STA. And that's, the net result there is a massive fluid loss and a diarrhea. And then you get the enteropathogenic E. coli. There the intermin, or the attachment, is by intermin. I'll show you a picture of the attachment factors and so on, and the virulent factors later on. So on the one side is the toxin is STA, and this one then the attachment factor intermin, and this the net result is problems with absorption and diarrhea. And the one that I would like to discuss a little bit more today is the Introhemologic E. coli. Now, they identify through the production of toxins STX1, STX11, or then the so-called verotoxins. The ST was also coming from, it's very similar to the toxins produced by the Shigella bacteria. And it also has got the attachment factor intermin. So again, like the previous one, the intermin is here. But the important thing is the production of the toxins STX1 and STX11. Now, this causes inflammation and then eventually destruction of the villi, and this then leads to a bloody diarrhea, and this is where the problem comes in. They implicated in enteric and systemic diseases in human, in white meat, specifically in pigs, then so-called oedema disease in pigs, and then diarrhea and dysentery in calves and goats. But I think the important, what I highlighted there at the bottom there, the E. coli serotypes 0157, is the major STEC. It was identified in 1977, implicated in human disease, and specifically E. coli diarrhea in humans. But the problem is this serotype is also very common in the intestine of cattle. So I would like to bring the thing out together, so we do get E. coli in animals, but it can be transmitted to humans, and that specific one that we're going to talk about today is the so-called Introhemologic E. coli, and then more specifically the 0157 there. Now on the left-hand side there's a nice picture of the various adhesions, things that's necessary, but also the some of the virulent factors that we do get, the outer membrane proteins, the different pili that we get, and then the toxins that can be produced. As I said, with hemorrhagic E. coli, STX, causing a lot of problems and causing necrosis of the villi, and eventually then a hemorrhagic diarrhea. But important there is, if you look at E. coli, not all of us will get E. coli just because we're carrying E. coli. I think the susceptibility of the host is very important. As I said, immune suppression, important factor that can cause problems. If you immune suppress for whatever reason, maybe you're not feeling well, you trying to cope with an upper respiratory infection, and then as a secondary thing, E. coli can come on board, and then you can be very sick. And then also the pathogenicity of the specific strain, that's also important to look at that. And then also the environmental predisposing factors. If you're in a clean hospital, there's no nosocomial infections there, then you will not get E. coli in a clean hospital, clean environment like in a hospital. But the possibility that you can get it, when you go and drink tea or coffee or having a nice milkshake at where there's a so-called petting zoo, the possibility is there that flies can transmit, from the animals, they can transmit this Introhemologic E. coli onto your table, onto your milkshake, and eventually you can then ingest this, and 2 or 3 days later you will be down with E. coli. So it's important, in animals and also in humans, we need to have this, need to look at if you look at the epidemiology, you need to look at the susceptibility of the host, the pathogenicity of the specific strain, and then the environmental predisposing factors. I think the people on board from the Western Cape, especially in the Western Cape, we do get a lot of E. coli mastitis, especially in winter time, and call it in the older days, when it was raining in the Western Cape, cows standing in the mud. It's not like that anymore these days. But that's also then, if the E. coli then enters the teat canal, that can cause severe mastitis. And it's usually, if a cow gets E. coli mastitis, it's a dead cow. So, that's the problem that we do have. E. coli enteric infections, I did mention this before, but again, I think it's important, that the organism, will survive the exposure to the gastric acidity, will go through the stomach into the small intestine, and there it will then adhere to the intestinal epithelium and then start multiplying. And while it's multiplying, then forming the toxins that's causing the problems there. Especially the newborn calf, as I said in the beginning, the very neonatal calf, the very, very young calf, 1 or 2 days old, a very, very young baby. The possibility there, if the little baby or the little calf or the little lamb do get E. coli, we do have a problem. It's sometimes very, very difficult to get him through that disease, and unfortunately, the morbidity is very high. In other words, the number of animals affected, but then also the mortality is very high. There's a lot of 40-50% morbidity with a 20-30% mortality. Why they actually get infected there? At that stage when that little calf is born, the intestinal tract is then designed to promote colonization and establishment of a normal flora. The animal is born without any flora that can help with the digestion and so on. And if this pathogenic E. coli can get hold there, it will take advantage of that stage, and it will then cause a problem there. Important thing that I think that when I looked at, putting this presentation together, and I got again in the, as a reference in one of the articles there, as that milk may contain a specific protective antibodies as well as other molecules that inhibit the colonization. So sometimes we do take, when there's diarrhea, we do take the milk away from that calf, and we only feed them water. So I think there's a lot of research that one can go and do there. Sometimes when we wean animals, usually a dairy calf is weaned at about 60 days. When we wean them, they do have a bout of day or two of diarrhea. And this is usually because we took that milk away, with those specific antibodies, and that can cause a problem there. This is again a lot of writing on the left, but I think if you look at the diagram there on the right, the important one there, you can see there where I'm circled with the red circle there, low immunoglobulin intake, the plasma proteins less than 5.5. That's usually where the problem starts. And it starts in the right top corner there. If there's an inadequate vaccination program, this is specifically now in dairy calves, but also in beef calves, induced calving, dystocia, any form of stress. But specifically, if there's an inadequate vaccination program, it can cause a problem, because there's, as you know, a calf is born without any antibodies. The calf must get the antibodies from the mother, and it is very important. There's a rule of thumb. The volume is about 10% of the body mass of the calf. So if the calf is weighing 20 kilograms at birth, it should get at least 2 liters of good quality colostrum from the cow within 6 hours. The longer you take, if you wait 12 hours or 24 hours, is too late. Those antibodies do get absorbed at a very early stage, and you need to feed that 10% of colostrum at least or within 6 hours after birth, and then of a good quality colostrum. So it's better to, if you want to develop a vaccine, not a vaccine bank, a colostrum bank, it's a good thing maybe to look at, collecting the colostrum from the older cows. That very first colostrum, the first or the second milking, collect it. You can freeze it. You can freeze it for up to 3 months, and then you can feed that. And I would rather take that instead of a young heifer that's calving for the first time. So that's what's actually on the left-hand side there. But if you see then, underfeeding is a problem, poor housing is a problem, poor sanitation is a problem. So this is again environmental factors that can play a role. And then high stock density. If you put your calves all together in one camp, the possibility is there that they can get a high level of exposure to the pathogens, and they will go down with one form of diarrhea. And eventually what we want to prevent is that increased risk of death on the right-hand side in the red block there. So this is, a nice summary of what can happen or what can go wrong or what can lead to diarrhea in calves. But eventually, this is what we talk about, the so-called FPT calves, failure of passive transfer. We need to do that passive transfer of antibodies from the cow, from that colostrum milk to that young calf. If we don't do it, we're going to sit with a problem, and that problem is going to be a diarrhea in the young calves because of E. coli. As I said, also, you know, a lot of these things are actually repetitive, but I think we need to understand there's not just E. coli. If you isolate E. coli, there could be some other viruses associated. So I think if you do have a dead calf, it's a good thing, you know, to do first of all bacteriology, but then also virology, you know, and then histopathology and at least a, you know, even electron microscope evaluation to see if you can identify the rotavirus and the coronavirus. Haemorrhagic coccidia, and again our friend Cryptosporidium may also be around there. Cryptosporidium, by the way, is a protozoa, you know, so we get viral infections, bacterial infections, and protozoal infections. Okay, then the thing is about, you know, we see it frequently, you know, the so-called calf raisers. They will go from auction to auction and buy all the calves that nobody wants. These so-called Friday auctions, you're going to buy yourself, not a cat in a bag, you're going to buy yourself a lot of problems, specifically with calves going down with diarrhea. We don't know, you know, what's the immunity of these calves. We don't know which organisms they carry in. They could be so-called Trojan horses or Trojan calves carrying highly pathogenic strains of E. coli. And then you bring it into your farm and then you do have a problem. So newly arrived animals, or conversely, you know, if you bring in an infected animal, it can affect your other calves there. So again, good biosecurity, you know, isolate that animal, keep it away for at least 20, I would say 30 days away from the rest of the animals, and make sure that you don't bring in some horrible organism there that you can't cope with. And then stress, you know, transportation, again, putting that calf onto the truck, taking it to the auction, from the auction back to a new farm. That transport is a form of stress. Overcrowding also is also some of the predisposing factors that can cause problems. So, okay, what do we need to do? There's a very good presentation, you know, from Dairy Standards. Jompie Burger and his team, they spend a lot of time on this, and I think every dairy farmer and actually everybody listening today should at least get one copy of this publication. But in this copy, you will see it's a code of practice for milk producers. There's a lot of attention paid to sanitation. You know, as I said on the right-hand side, poor sanitary hygiene may result in massive fecal contamination of the external environment. That can end up in your clusters in the milk machine, and eventually it can end up in the milk. And this is not, you know, most definitely we don't want to have it in the milk. So over time, if there's poor hygiene and poor sanitary principles or sanitary principles not applied, you will end up with a problem with possibility of a pathogenic E. coli going through the dirt, causing diarrhea, first of all in the calves, eventually E. coli mastitis in the cows, meningitis in the lambs, and some pigs. If there's pigs on the farm, they can also be infected, can cause a lot of problem. And then also, you know, if as I said earlier, you know, this is a facultative organism, so wherever it finds an environment where it can survive, it will survive there. And if it's especially in a damp area, you know, if it's a wet area, it can survive there for much longer, and it's the consequence will that it will be spread. The infection can then spread. Okay, this is a nice picture I got, you know, just to summarize our little friend, the little piglet there. You can see at number 1. So from number 1 is the ingestion of the pathogenic E. coli. It can be from soil, it can be from water, it can from the other animals. You know, there's 1 or 2 animals with diarrhea around there, especially piglets. You know, when you keep them together, they're usually in a group. Compared to dairy calves, you know, dairy calves usually in separate little hutches, or, you know, somehow you need to separate dairy calves. But then after ingestion, it will colonize in the intestinal respiratory, or in the respiratory, or some of the other mucosal surfaces. As you can see there, you know, with the fimbriae there, it will attach to the villi of the intestine, and then it do have the ability to actually pass through the mucosa, and then eventually end up in the in that whole process, then producing the toxins. and then eventually it will end up in the blood vessels, and then causing the so-called septicaemia, or the persistence, and then again multiplying, and all the time producing these toxins, and then going to, I think number 8 will come before number 7. So you will get a localized infection, and then you will get a shock and death, as the end result there. But it also has the capability, and we're not going to talk about immunity and immune factors and so on. But this bacteria also, if you can see there at number 4, has got the ability to, to the resistance to the complement and also the phagocytes in the immune system. So that's a nice summary of the pathogenesis of the organism. Okay, the clinical signs. I think I've mentioned it now several times. Acute disease. Acute disease. Sometimes you will just see a dead animal. I think it's important to do a post-mortem on every dead animal. It should be done by a qualified person, like a veterinarian. Veterinarian will take the samples that they do need to take, and from there they can actually make a definitive diagnosis. We can't just say, diarrhea is part of raising calves, because this diarrhea that you can see there on the left-hand side there, that's the back end of the calf, if you haven't recognized it yet. But that diarrhea is highly potent, and it can cause disease in humans also, and that's not what we want. But usually that calf will be listless, it will be dull, disinclined to drink, it will refuse water or milk or anything, and then eventually it will show a profuse watery diarrhea, which usually is whitish yellow or grayish. Again, you can't just say, if it's a putty-colored diarrhea, it's salmonella, and if it's whitish yellow, it's E. coli. I think don't go there. We can't diagnose on that. That's just giving us indication, that we do have a problem. Okay, and again, what I said, the association with some of the other organisms. Usually, if you do get diarrhea a little bit later on, it could be, at week 1, week 2. Week 1, I would say most probably it's E. coli. If it's week 2 or 3, most probably E. coli, together with some of your other organisms like your Rota and Corona and Pasteurella and so on. So, okay, the diagnosis, again, I'm not going to look at detail there. There's what we do in a lab. I would say that's the old way that we did it. I think these days with genome typing and so on, we can go into a lot of more detail. And then also the screening test that's available, the lateral flow test as an example there at the bottom, where you put a sample of the diarrhea in the little chamber there, and then give it some time. That will then, there's a binding then between the antigen and the antibody. It will give you a discoloration there. And it can give you an indication that we do have E. coli here. But it's not giving you an indication, this is a specific test for E. coli 0157. But a lot of these tests don't give you a specific indication. It just tells you it's E. coli. And then I want to say almost, so what? We need to identify is this really a terrorist, or is it just a person wandering around, and that doesn't have anything to do. So I think the important thing is, rectal swabs, as I said, there's less satisfactory. But yeah, we need to diagnose this organism and make sure that we do have identify them. And then there on the right-hand side is a nice photo of your Cryptosporidium. But you can get Campylobacter, Salmonella, Clostridium perfringens type A, B, and C, your Brachyspora, Cryptosporidium, Eimeria, Isospora, all your protozoal diseases could be some of the differential diagnoses that we need to look at. But I think the first thing when you look at the diarrhea at calves, the first thing that we need to look at is the possibility that it could be a so-called nutritional scours, maybe an overfeeding with milk. As you know, the milk should not end up in the rumen. The rumen is underdeveloped in that young calf, so it should end up in the abomasum. You must make sure about the feeding, the way that you feed these calves and so on, that milk doesn't end up in the rumen itself, because that can cause a diarrhea without E. coli there. Okay, the control. I'm not, again, not going to discuss this for time, to look at time. I need to push a little bit here. The quality and the quantity of the colostrum. This is an NB. If this was given for a student, this is for exam purposes. The quality and the quantity of the colostrum is very, very important. and then also, you know, the temperature that you feed it at and so on. And then also very important to prevent the disease is the vaccination of pregnant animals for E. coli, because that little calf is born without any antibodies, and it can only get the antibodies from the mother through the colostrum. The treatment, again, you know, I don't think it's our place today to discuss this in detail. Oral antibiotics is sometimes advocated, but I think don't reach for the bottle, and please don't put injectable antibiotics in milk. We're going to cause antimicrobial resistance, and that will cause a problem later on. If you really want to use a antibiotic and you've already developed or you're already the cause of an antimicrobial resistance, you will end up with not, the animals will not be able to survive, and we will have a dead animal. I think if you look at the recent developments, you know, there at the top, I've said something in 1982, as I said, Escherichia E. coli 0157 was, I think in 1977, I said earlier. In 1982, the first outbreak of haemorrhagic colitis associated with undercooked meat was, I think it was in the US that identified that. But what we've seen over time now, from 1983 up to now, with the recent molecular analysis, you know, we've seen that the 0157 strains are apparently getting more and more virulent and causing more and more disease. So that brings me to the second part of what I would like to discuss. That these underfindings underscore the need to be vigilant for these pathogens and to apply One Health approaches to minimize the potential, because this is a zoonosis. It can be transmitted from animals to humans and also from humans back to animals, to minimize that. So if you look at the whole One Health thing, and again, not going to discuss all the detail, but you know that One Health is people, the land on the left bottom there, you know, wildlife, farmed animals, domestic animals, and then water on the right-hand side. All of these can play a role. Now, I've got this nice diagram in an article looking at the different factors that can play a role, and I would like to take you through this quickly, you know, to conclude the part of One Health. So the important part is there in the middle, you know, important part in the middle. It all starts there with the feces. Contamination with feces, you know, whether it's in your on your hands, on your boots, or wherever, you know, if it's a dairy farmer, or even us, you know, when you go out, possibility that you can pick it up anywhere out there is there. You know, a person could be, you know, maybe, yeah, let's call a spade after going to a toilet and not washing your hands. The possibility is that they can transmit it in that way. So the fecal oral route is, you know, that's the main route of infection. That can also be with contaminated produce. You know, there's a lot of dairy farmers using slurry dams. Usually in the slurry, you know, the fermentation reactions will have an effect on the E. coli, but it's a possibility that you can actually infect your pasture with E. coli with water coming from the slurry dams is also there. Infected individuals are highly contagious. You know, whether it's a calf or whether it's a human, stay away from that person or stay away from that calf. If you do need to work with the calf, use gloves and, you know, get rid of the gloves afterwards. Otherwise, if you do get in contact with your hands come in contact with your mouth, you will know that you have been in contact with E. coli in a day or two from there. Okay, then the important one, going to the next level there, the transmission. As I said, irrigation can, soil, water, undercooked meat, you know, most definitely in abattoirs, undercooked meat, you know, your people eating raw meat and so on, the possibility is there that you can pick it up there. And then one of the things, you know, I think I've said in the previous discussion also, all milk need to be pasteurized. You can only drink pasteurized milk. I think it's in inverted commas, it's a sin to drink unpasteurized milk because the possibility is that you can get E. coli there. All milk will be tested, you know, at arrival at the milk buyer. All milk will be tested for E. coli. So the possibility, you know, that you can introduce E. coli into the food chain, specifically through milk, is almost zero. But yeah, pasteurization fortunately will kill the organism. And then insects also can transmit it. I will get back to that just now. And then humans, you know, the human carrier, possibility that a person could be a carrier. It's immune compromised and it could be shedding this organism all the time, and all the time maybe working with food or working in the dairy itself. And I think it's a good thing by maybe people working with food, you know, that they do get tested from time to time to make sure that we don't transmit the bacteria. As I said now several times, the reservoir of 0157 is usually it's ruminant animals, usually your young calf, but it could also be your adult animals. And this is, you know, this is a seasonal type of thing that we see. It could be temperature, it could be, you know, there's a lot of other reasons that can contribute that it's a seasonal thing. Then also, you know, the single strain, a specific, or not a virulent, a pathogenic strain, can survive for quite a long time on a specific farm. So if you do buy young calves from a farmer, again, by security, isolate your animals and then test them before you introduce them to the rest of the animals. Because this organism can survive in various forms, you know, outside the animal and also most definitely inside the animal. Okay, if you look at sub-adult cattle, you know, that's from 2 months to 2 years. They tend to shed a little bit more. You know, the biological reason or the basis for the seasonal or age-related peak shedding is I couldn't find anything in the literature, but it's, you know, so there's some more research that we can do. But it could be, you know, with day length itself. It could be with affecting hormone levels, you know, hormone levels in the cows and so on. And then also the seasonal presence, you know, in KZN and in Eastern Cape, where we've got the seasonal calving patterns. Those calves, there will be more calves around, and then the possibility is there that we will have more E. coli around there. Okay. Both sheep and cattle are characterized host of your 0157 infection of you know, the haemorrhagic E. coli of that they could be the carriers there. Uniquely, they colonize the few centimeters of the last few centimeters of the intestine, and I'll refer a little bit in more detail about fly transmission. You know, if there's flies around, I see them as enemy number one. Get rid of the flies. They can transmit this disease. They can transmit viruses. They can transmit a lot of things. There should not be a fly on a dairy farm. There should not be a fly in a restaurant. There should not. Yeah. Sometimes I ask the question, why did they create flies? But yes, we do need them for some reasons, but most definitely not on a dairy farm when there's calves around. Okay, other domestic animals, you know, deer, like your wild animals, birds, swine, rodents, rabbits. You know, if there's a little petting farm where you can go and play with a nice little furry white rabbit, the possibility is there that that person can pick up that E. coli there from that little rabbit. So please, you know, if you do go there, ask the people, you know, the little children, go and wash their hands again to make sure that they don't pick up the disease and or transmit the disease. Non-ruminant animals, horses, dogs, they looked at pooled bird feces, pooled feces from flies. They did pick up the organism there. Domestic and feral swine, you know, your war dogs as an example, possibility is there. And then rats. You know, again, rats should not be on a dairy farm, or it should not be anywhere. As an example, you know, this was a specific article associated with rats, with a dead rat in the feedlot water tank, and suddenly there's in animals that in a feedlot, you know, they 220 days old, and suddenly there's an E. coli outbreak of E. coli 0157 in that feedlot. Where did it come from? A dead rat that was carrying E. coli, and it went into the water tank, or it was alive, and then it went into the water tank, and then it contaminated that water tank. So as you can see, you know, there's a lot of things that we need to look at. And then wildlife, I already referred to wildlife, already referred to birds. You know, there's some specific article that I looked at, even your tadpoles and then your bullfrogs. They actually, you know, specifically in stagnant water, in running water, in the river, you know, running river, it won't be a problem, most probably. But in stagnant water where you do get tadpoles and so on, the possibility is there that they can also host this E. coli and then contaminate the water. Yeah, and I decided to put this nice picture of our enemy number one there. You know, if you look at the fly population, one fly, one domestic fly, Musca domestica, can cycle in a life cycle of a very short lifespan. She can lay quite a lot of eggs, and those eggs will become larvae, and the larvae will eventually become a new fly. But they can, as you can see, they're sucking food or sucking, ingesting diarrhea or whatever, from diarrhea, can eventually contaminate, and it will be then cross contamination to the next animal. And then also important is actually our friends, the little dung beetles there at the bottom, but they've also isolated E. coli from the dung beetles. So yeah, we do have a problem. So if you look at the environment itself, at cattle showgrounds, they actually picked up 10 months after the show, they still picked up E. coli, pathogenic E. coli in that environment, on a specific farm for quite a long time, contaminated water, and then, as I referred to already, the water troughs. So that's in the environment itself. So if you look at the whole One Health thing, as I said, One Health is land, people, and then the animals, and then in between the pathogens. There's a little diagram about the One Health principles. But this, this whole thing that I showed you to you and shared with you, clearly shows the relevance of the One Health concept. And I think this is then, as I said in the bottom two lines, the convergence of humans, animals, and environmental domains. When these three things come together, we do have a problem, and the possibility is that E. coli can play a role there. I think if you look at on the farm to prevent this, if you look at the One Health approach to diagnosis, prevention, and control, again, we haven't got time to look at this in detail, but if there's an outbreak, we need to look at these things, the factors that's contributing to the disease. But I think there's a lot of probiotics on the market, that can prevent this. There's antimicrobials, speak to the veterinarian, or in a human case, speak to your medical doctor. Disinfectants, where maybe we can, during question time, discuss that in a little bit more detail. And then your bacteriophages, there's a lot of development in that specific field where they can develop a bacteriophage that can go and attack this E. coli. And then we need to look at the vaccine strategies. Maybe we can also discuss that in a bit more detail if there's some time during the question time. But I think is, to summarize this slide is, and I'm going to read that, we need a coordinated multidisciplinary effort. So coordinated, yes, animals can play an important role to host this organism, and then could be almost like the, providing this all the time and pushing it into the environment. So dairy farmers need to look at this, veterinarians need to look at that, but most definitely also in a multidisciplinary environment, we need to just not to point fingers, but need to look at it also from a human medical perspective and see how we can approach this. We need to understand this whole life cycle and this whole One Health approach, and then to integrate the epidemiology, the pathogenesis, and then also the pathophysiology. Because, as I said now several times, we need to prevent, we need to control, and we need to treat this zoonosis, because it's a huge problem. If a little baby do get this, we can have a funeral, and that's not what most definitely what we want. So yeah, in conclusion, I think I would like to conclude and say, there's some important food and waterborne pathogens of humans. E1 Introhemologic E. coli 0157, specifically 0157, but there's a long range of other E. colis also that can play an important role. But again, also there's some good E. colis also. If an E. coli can be good, some good E. colis out there. It's not necessary, again, if you diagnose E. coli with a rectal swab, that it's E. coli as the cause of the problem. There's many serotypes there of the non-0157. Human infections is usually from diverse exposures. That could be contaminated food, contaminated milk, especially, from bovine origin. So again, we don't need to go and slaughter all the animals out there. We do need the milk, we do need the meat. We need to work together, in a multidisciplinary team to counter this. And then to look at the contaminants and the ingestions of other food. So in the end is my last part here. I think cattle and also other animals, they fit the definition of a reservoir host. But the instability of cattle colonization, coupled with the evidence of stable environmental contamination, suggests that this is a zoonotic disease, and that can infect people, and this is not what we want. But I think in the end, as we involved here with a complex environmental host ecology that directly affects the likelihood of hemorrhagic or enterohemorrhagic E. coli 0157, and we need to look at all these things and address it one by one when we do have this problem. Adele, yes, that's a lot in, I think, 35 minutes, but that's what I do have to say on E. coli this morning. Thank you.
Thank you so much, Chris. Wow. Yeah, I wonder how often E. coli is the culprit of the so-called stomach bug. Or, you know, if people say you must come and fetch your child at the nursery school because there's a stomach bug going around. I wonder how often E. coli is the culprit. Thank you very much for that very comprehensive presentation. I really appreciate it. To the audience, now's the time for you. If you have any questions, please place them in the question bar so that I can ask Chris. Chris, from my side, what I would like to ask, or the way that I've taken your presentation, is that it doesn't seem like we're going to get rid of E. coli. However, it's very important that the control at the farm in terms of pest control, the flies, and cleaning and all of that. Is there something, you know, chemicals or stuff when cleaning that you can utilize that can help to prevent this?
Adele, yes. I think first what you said now is we will not get rid of E. coli. You know, we can counter it. You know, there's a lot of advancements in the development of vaccines. But I think at present, you know, what you said now is you mentioned two names, cleaning and sanitation. You know, you can't just only clean and not sanitize, and you cannot sanitize if the place is not clean. So, you know, to use a sanitary agent and put it on top of a lot of feces and so on is not going to sanitize the place. So I think first of all is, you know, sanitation is one of the, if you look at the epidemiology, you know, we're looking at the host factors, we're looking at the pathogens, and we're looking at the environment. As I said, you know, the mother cow needs to be vaccinated, but then very, very important, the colostrum, the transfer of colostrum to prevent getting the diarrhea there. But then the sanitation itself, you know, sanitation is one of those things. We've seen it now with COVID-19. You know, I'm looking the other day, looking at one of these sanitary stations at the entry when you go into the malls. Some of them standing there broken. Nobody is using it anymore, you know, because we think that COVID is gone. COVID is still there. E. coli has been here for 140 years. So E. coli will not, in the next 100 years, will not go away, because it's got the ability to mutate and to counter whatever we can throw at it. So we need to look at a clean environment. We need to have separate calf hutches, you know, in a dairy farm. We need to have good ventilation, and then also bedding. You know, you can't just keep a calf in a dirty camp, you know, where it was previously a pigsty, and say that calf will grow up and will not have a diarrhea. And then the basic biosecurity. I think the role of people. You should not allow people at your calves, you know, because maybe that person was at his own calves this morning, you know, and you know what people do, and especially when you get visits to the farm. I think children should not be allowed into the calf, because into the calf camp, because one of those children could be a carrier of one of these pathogenic E. coli and introduce it. What children like to do is to put their hand in the little calf's mouth, you know. So the management of people, the management of animals, you know, the host factors, as I said, to isolate animals, and then the pathogens itself. But then if you look at disinfections, you know, there's several things that one need to look at it for disinfection. One need to look at the active. You know, water is also a very good disinfectant, but up to a certain level, you know, up to a certain level, it will get rid of many organisms out there. And many organisms doesn't like water, or don't like water. So yeah, but we need to look at the active. You know, is it the quaternary ammonium? Is it a acid and alkaline or whatever? We need to look at it as a registered product. You know, products should be either registered under Act 5 of 2008, if it's for inanimate objects, like for tables and surfaces and so on. If you're going to use this product, you know, in environment of animals, it should be registered under Act 36 of 1947. Very, very important thing, read the label, read the exposure time. You know, some of these things have got an exposure time of 10 minutes. Some of them as short as 1 minute. So, you know, after 1 minute, it will be effective and it will be will kill the organism. Some of them only after 10 minutes, the organism will be dead. The concentration, if it says 1 to 3 dilution, don't save on that and then make it, you know, 104 and 105 to see, you know, because the product is expensive. That's the last thing that you need to look at. Then you need to look at the log reduction of the organism. You know, it will show you there it's a log 4 reduction. That's as an example, 99.99% of the organisms will be killed. You do get, as an example, of FND, foot and mouth disease, the products on the market should have at least a log 4 reduction. A log 3 and a log 2 will not do what it needs to do. And then the stability and, you know, the water quality and so on, what you're going to use, also to look at that when you look at sanitation. So I think it's important, you know, the very, very last thing is to look at the cost. Price is what you pay and value is what you get. So, you know, use a good quality product, a registered product, and then you can sanitize the environment, and then you will prevent this problems. But that should be in all places. You know, it should be still using it at restaurants. There's some restaurants around Pretoria, you know, there's nice cows around us, but it's important, you know, to sanitize your hands when you go in there and sanitize your hands when you go out of that restaurant.
Yeah, and I think also important, you know, work closely with your chemical company to ensure that you get the proper material safety data sheets and make sure that what they say they're registered for, that they can provide you with the paperwork, you know, that they are really registered to do what they say on the material safety data sheet. You spoke about vaccination as well. Can you explain a bit more of where, what, and how, and you know, do you do that with your vets? And you also mentioned that it may be seasonal. So around vaccination, can you just explain a bit more on that?
Yes, I'll do. I think there's very good vaccines available these days, Adele. There's vaccines available. I'm not going to mention trade names now, but it's, you know, there's certain so-called monovalent vaccines. I think the monovalent vaccines, in my opinion, haven't got a role to play here. One need to look at the combination. As I said earlier, you know, the vaccine should at least contain a rota and a coronavirus. It should contain the E. coli. It's usually the K99 E. coli, and then, you know, something else like Clostridium perfringens type C that also can cause a haemorrhagic diarrhea. And then what you need to do is usually, you know, if you vaccinate your heifer for the first time, the pregnant heifer, you can vaccinate at dry off, and then again about a month before calving. So because it's usually a killed vaccine, you should have a double boosted vaccine. And then usually after that, you can only do it once a year, about 2 to 3 weeks before calving, during the dry period. You know, the best time is to during the dry period, when that cow is developing or forming all the antibodies in the immune system and so on, you know, to vaccinate your animal in the dry period. And then to make sure, you know, that good quality, then you've got a good quality colostrum, and that can be the volume, and then the time is very important, you know, to get it over to the calf. If you look at, there's some development, you know, with all the genomic work and so on, development where they're actually looking at certain vaccines that can, and that's in development phase, that the vaccine can, you know, there's immunoglobulin G antibodies specifically against the intermin, as an example, that's in your Introhemologic E. coli. So that will reduce the bacterial shedding, and, you know, because of that, we will get a reduced shedding from animals, and that, you know, contamination of the environment is then less. So I think, yeah, I'm not sure if there's a vaccine available in humans, but yes, there's antibiotics available, and there's a lot of other things available if you do have the problem. Very good quality antibiotics, but I think rather, you know, speak to your veterinarian or speak to your medical doctor before you start using any antibiotics.
Yes.
With suspected E. coli infection.
Definitely, definitely. Yeah, I think for me always, specifically now going back to where you were talking about, preschools, going to petting zoos, or if a preschool determined that they've got, the stomach bug, I always say to people, I wish they can realize there's nothing like a stomach bug or a flu. It's definitely something that you took in, or cross-contamination took place by hand, causing a person to have diarrhea. So I also feel something, especially for the environmental health practitioners online, when they go out to the farms, the pest control, and also at the tea gardens that you've mentioned, where there's a lot of flies, specifically within environments that it's close to farms, that they definitely need to look at that as well. I've got one question. I've got two questions here. The first question is E. coli also prevalent in live chickens?
I think they can become contaminated. They do a lot of things. If you look at the raising, I'm not a poultry specialist, but from my long ago knowledge, from as a veterinarian, they can become infected, and then when you slaughter them, it can actually contaminate. It can be in live chickens and then eventually end up in the meat. So the possibility that you can pick up E. coli is there. So the best is, buy your chicken meat, buy it from a reputable source, and then, you will not have a problem there.
Absolutely, absolutely. Yes, quite a long question. I think I'm going to break it up. Firstly, Diane says, thank you very much for the good presentation. She says here, for these farm animals that are symptomatic for E. coli infection, are these E. coli perhaps antibiotic resistant or multi-drug resistant? And if so, are these the ones disseminating in the environment? Is antibiotic use regulated on farms, and could this be contributing to antibiotic resistant strains?
Okay, Adele, have we got another 2 hours?
No, not really. I need your short, sweet answer here.
Okay, I'll try to quickly answer it. Yes, Diane, I think this is a very good question, and I would like to discuss it in more detail later on. So there's my email. You can contact me and we can discuss it. I'm now also involved in the Ministerial Advisory Committee on Antimicrobial Resistance, and most definitely we will be looking at this. The regulation is there. Yes, antibiotic use is there. There's two types of antibiotics. We get the so-called over-the-counter antibiotic. That's like your tetracyclines and certain your sulfas and so on, that a person can go and buy at the co-op, or it can be bought at the co-op, and can be used by a farmer, or it can be used by anybody. But we need to make sure, your tetracyclines, your sulfas, and as an example, I think there's already a resistance against that in many, your respiratory pathogens and so on. We do have a problem. And then we get the Act 101 medicines products, that's SAPRA, and that's regulated by Act 101. Those products are usually from Schedule 3 up to Schedule 4, 5, and 6, but usually your antibiotics will be a Schedule 4, and that can only be prescribed and actually used by a veterinarian. If the veterinarian is au fait and is knowledgeable about what's happening on a farm, he can prescribe that product. But, to loosely just to give an antibiotic to a person, say, I've got diarrhea in my calves, and do you have some antibiotic for me? I think that's, again, that I won't say you need to go to prison for that, but, I don't think you should be doing that. So veterinarians, medical doctors should not just prescribe antibiotics, because that can eventually end up, with a resistant O157 ending up, as an example, on a farm, and it can contaminate water, it can contaminate the streams. Eventually, downstream, it can infect people and infect another herd and so on. So yeah, this is a vicious cycle. The overusing of antibiotics. I think we need to address it. And again, what I said to a person the other day, you can't cure everything on a farm out of a bottle. You need to look at the host factors, you need to look at the environmental factors, and then most definitely you need to be 100% sure again, who's the enemy, who's the pathogen. Is it just a normal E. coli or is it a pathogen E. coli? And then you need to look at management on the farm. A lot of these things can actually be sorted out with good quality management. So, yeah, I don't know if that's a short enough answer, but yeah, that's what I will say.
Thank you. I'm sure of it that answers Diane's questions, and thank you for the answer. Yes, unfortunately we don't have more time for more questions, but Chris, thank you so much for your time. We really appreciate it and all your valuable input. I think once again, this has shown, like you said, this is a multidisciplinary approach, you know, from the farmer right through to whoever is preparing food for people. You know, a lot of people is involved, and we have to be aware of that we can prevent it. And we have to make sure that we look at the preventions on the farm in terms of the hygiene and everything that you've mentioned. And with that, I just want to say to everybody that the webinar will be recorded and it will be available on our website. If you go to www.foodfocus.co.za, you will, on our homepage, see that there is a bar at the top that says resources, and all the previous webinars from the DSA is available there to download and to have a look at. Also, we've got a newsletter. Please make sure to sign up for the newsletter so that you get the news if we are hosting other webinars. The DSA is hosting another webinar in November, and as soon as we've got the topic, we will make you aware of it. Please also note, in the presentation, Chris spoke about the Dairy Standard Agency, the guidelines and codes of practices that they've got. So if you go to dairystandard.co.za, you will be able to download that document there that he referred to. Please note, after the end of the webinar, there's a short survey. Please take the time and just answer the survey, because in that way we make sure to provide you with more webinars with the topics that you would like to hear more about. And with that, thank you so much for your time. Hope you enjoyed the webinar, and until next time, have a blessed day. Bye-bye, everybody.
Thank you.